So, continually, however, we still hear from funding agencies, particularly some outside NIH, that we will learn something. But the good question to ask is what? Because we don't really learn things that we need to learn in science. Now, what are the difficulties in developing a successful HIV preventive vaccine? Well, I've also alluded to some at this point. We cannot use attenuated live virus nor whole killed virus, so we use subunits. We have a limited animal model. I don't think it's a bad animal model. I think the primate model system for HIV vaccine development is actually pretty good. But it's numbers that are the problem. To this day, there has never been made available adequate primates for an adequate number of investigators, and there's a bias in which way those primates will go. I believe this is a problem that is solvable by NIH and or the Gates Foundation, because, bear with me, if you do an experiment in primates with 5 monkeys or 4 monkeys, which is usually the case, you get results that are interesting. What happens? You have to repeat them. Each one of those experiments takes 1 year, 2 years, 3 years. So you reach a decade pretty rapidly. And if you change anything, the adjuvant, which suddenly a company comes along and says, you can't use that adjuvant anymore, which happens to us. We had a very good adjuvant, QS21, and then GSK said, no more, they had control of it, and you can't get it anymore, so you have to go back to ground zero again. Because every time you use a new adjuvant, you start from scratch. There is, as you all know, enormous variation in the virus. There's enormous variation in a lot of viruses. I don't think that's as big a problem as been made. It's a problem that is famous because it's simple and everybody mentions it all the time, but I believe we've moved past that. Another problem is that HIV very, very quickly is seriously impairing the immune response. So things to do something about it have to happen very quickly. But these last are the most important difficulties, and this room is all well aware of it. As a retrovirus and integrating, it seems to me, and the rapid immune impairment, demands that we approach sterilizing immunity, or at least get close to it. To my knowledge, that's never been done in the history of vaccine development. At least nobody knows that they ever achieved sterilizing immunity. And just think about polio. We're all vaccinated. You drink a vial of it, we all get infected, or almost all of us will get infected. But after a few weeks, we clear the virus in the gastrointestinal tract before it reaches our brain or our spinal cord column. So therefore, you don't get disease. You clear the virus ultimately, usually, completely. There's another point, and that's immunity has to be sustained. And it's often forgotten about because we all do vaccine experiments. You challenge an animal after you have a peak of immunity. So in other words, you vaccinate, you follow the immune system, you reach a peak and you challenge, you say, oh boy, I got protection. And it's heterologous challenge and it looks great. The problem has been, if you're focused on envelope-based vaccines as we are, that almost all envelope-based vaccines have a sawtooth response. That is to say, you last 2 to 4 months and it crashes. The B cells that make antibodies to envelope must be short-lived or have some kind of problem with the envelope of HIV. I don't think that's true with many animal retroviruses. The immune response is unfortunately short-lived. We must solve the problem. We must develop the right kind of adjuvants or something else, maybe the right cytokine that prolongs antibody response, perhaps prolonging B-cell survival. This is actually a major focus of our institute by a senior colleague, George Lewis, is back to studying the basics of B-cell biology and memory B cells. So, finally, I would say the best approach must include a mechanism that leads to a block of entry, once again, into cells, or that kills infected cells early and efficiently, likely achieved with antibodies, and suggests that an envelope-based vaccine is at least a part of any successful vaccine of the future. The difficulties has been that an envelope-based vaccine, as I've already alluded to, has produced type-specific rather than broad immunity, and short-lived, which should be on this slide. And the question is, can we get around the problem? Can we make a vaccine be broad? Can we make the memory last? Well, one approach is to look at what sequences of HIV GP120 envelope are conserved and functionally required. The GP120 of HIV interacts ultimately with CCR5 after binding CD4. To us, it suggested an approach for a preventive vaccine because the interacting portion of GP120 with CCR5 is highly conserved, functionally required. When you get infected with HIV, almost always it is targeting CCR5. It's rare that CXCR4 is used to begin infection. So this region is functionally required and conserved, but the region is hidden. It's hidden in an inner domain of the envelope, covered by what is called the outer domain, and that enclosed by a carbohydrate shield. Further, the envelope is in a kind of mobile, dynamic state. So this has been a serious problem in developing antibodies that would be specific to this region. However, in thinking about getting around this problem, there is a large, as indicated in the bottom of the slide, conformational change in GP120 that is quickly induced by its binding to CD4 and the subsequent constraint of GP120 while exposing this site. The slide is somehow not complete when I just modified it. So in other words, if you have it bound to CD4, that previously hidden site can become exposed and the envelope becomes constrained. It's no longer in that dynamic state. So this was a concept we began to pursue about 7 years ago in trying to develop an HIV preventive vaccine. That we would take GP120 in a form that is bound to the binding region called D1, D2 of CD4. The envelope would no longer be wiggly, but would be constrained in this transitional state, and the region of the interacting portion of the envelope that finds CCR5 would be exposed. This slide is taken from, I think it was the last paper published from Don Wiley before his unfortunate death, from Harrison and Chen and colleagues. And the left, as you look at it of the slide, shows their image of GP120 before binding CD4. And they describe the change after binding CD4, shown on the right, as the largest change they knew of in biology with protein-protein interaction. Concentrate in the region at the bottom in red and just to its side. The gray region is the so-called outer domain. But after binding and looking to the right at after binding CD4 and you look to the right, you see the major positional changes and structural behavior of the V1, V2 stem, and you have this region that's called the bridging sheet, which is the region that you have interacting with CCR5 that is enormously modified. So bound to CD4, we have a new form of the envelope and one that exposes this site. So going through the stages of HIV entry into the cell, this schematically depicts the GP120 interacting with D1, D2 of CD4. Then we just drew the change in the envelope from the circle to bean shape and binding now to CCR5 preceding the fusion events. And so this is the vaccine that we use in the Institute. However, not exactly like that, because there's a loop of 20 neutral amino acids that link the binding region of CD4 to GP120 that's not exhibited by that cartoon.